I'm Daniel Levine, and this is the Bio Report. Ischemic stroke remains one of the most devastating and resource-intensive emergencies in medicine, with the physical removal of a clot, the current gold standard of care, still leaving more than half of patients with poor outcomes at 90 days. Revalesio is developing a next-generation oxygenated saline infusion designed to protect the brain during and after clot removal by stimulating mitochondrial function, reducing reperfusion injury, and limiting the spread of damage to surrounding tissue. Revalesio President Greg Archambeau discusses how the company's experimental therapy may reshape the trajectory of stroke recovery, shorten hospital stays, and change the economics of stroke centers. Greg, thanks for joining us.
Yeah, Danny, thanks for having me.
We're going to talk about stroke, neurodegenerative diseases, and Revalesio's efforts to develop an unconventional therapy that targets mitochondrial dysfunction and neuroinflammation in these conditions. Perhaps you can start by explaining what role mitochondria play in brain health and what happens during a stroke to cause mitochondrial dysfunction.
Yeah, sure, Danny. I think, mitochondria, I think most of us are familiar with the role it plays as the power plant of cells, but it's important in brain health with regards to learning and memory. Mitochondria supplies the energy to neurons, and neurons consume an enormous amount of energy. So small drops in energy can really impact the efficiency of mitochondria and trigger neuronal stress. And so this plays out in brain health, but it also plays out throughout the body, given the important role that mitochondria has. And then I think your second question was about specifically stroke. Is that right?
Yes.
Yeah. So in our setting, we're looking at ischemic stroke where there's a clot and that causes insufficient blood supply, oxygen supply to the mitochondria. And so it's kind of that same story. You're going to have less energy supply going to those cells, those key cells within the brain when they're under attack. And so because the mitochondria is operating in a kind of an area of deficit, you're going to see more neuronal cell death. You're going to see inflammation, and ultimately that's going to result in the loss of brain tissue and stroke, and then ultimately disability with the patients later on.
Your lead experimental therapy, RNS-60, is in development as an adjunctive treatment to endovascular thrombectomy, the catheter-based removal of a clot. This is done to prevent damage from reperfusion injury. Can you explain what happens during a stroke and then when blood flow is restored, how that causes additional injury to the brain?
Yeah, so if you have an ischemic stroke, you've got a blockage, and you alluded to what, really the best standard of care right now is you could get a thrombolytic drug, which is meant to try and dissolve the clot. But really the gold standard is mechanical thrombectomy, where you're removing that clot because you have to reestablish blood flow to that area of the brain that was served by that vessel, that artery that is now blocked. So when you remove that clot mechanically, you're right. You mentioned about reperfusion injury. You have the reestablishment of blood flow is essential, but in doing that, there's some damaging products that can go downstream, like O2 radicals that have been created by that compromised mitochondria, those compromised cells. And that can kind of feed into increased inflammation and really exacerbates the primary damage that was caused by the blockage.
What is RNS 60 and how does it work?
So RNS-60 is a solution that we've developed based upon parameters associated with fluid physics. We saturate saline with oxygen to about 60 milligrams per liter. And so you can think of it as an oxygenated solution, but we've done research showing that it has benefits beyond what's just provided by the oxygen. There's additional cell signaling that occurs that helps with the stimulation of mitochondrial function and also helps maintaining mitochondrial structure in these conditions or diseases where the cells are under stress. Now, um, we are just a few weeks away from having a publication in Muscle and Nerve that really highlights and shows some great photos of the deficiency in mitochondrial structure. This is actually in an ALS TDP-43 mouse model. And then the maintenance of that mitochondrial structure if RNS-60 is dosed in that model. It's a great piece of work and looking forward to getting published.
Well, how well understood is the mechanism of action of RNS 60 and what's happening at a cellular level when someone's treated with it?
Yeah, so we know a lot about RNS-60. We've got over close to 2 dozen publications, and we know that it engages pro-survival pathways in multiple cell types. So that's an AKT pathway, for example, in neurons. But we've also seen pro-survival effects in oligodendrocytes and other cells, glial cells, for example, that play a role in preventing inflammation. So we understand a lot about RNS-60 as it relates to both an acute setting of a condition like with stroke, but also in a chronic setting where you're talking about ALS or Alzheimer's.
What preclinical or translational data around mitochondrial biogenesis or ATP production or inflammation made you feel that this is more than a placebo?
Well, we've generated data at multiple different labs, and it's the consistency that we've seen with response to the mitochondria that really we know that it's more than a placebo. And I could point directly to the preclinical work that we've done in stroke, both in a rodent model and a primate model, where we saw a 50% reduction in brain volume loss in stroke models. And then, of course, we've now seen that in a phase 2 study that was double blinded. So we know that the effect is more than a placebo. It's really a potential substantial change to the standard of care for the treatment of a ischemic stroke.
Let's talk about the phase 2 study. Who are the patients, and when they received RNS 60, what were the primary questions?
So the patients that we looked at were those individuals that had a major stroke. So we were looking at individuals that had what's called a large vessel occlusion stroke. These are candidates for mechanical thrombectomy, where they, as we discussed prior, they go in and remove the clot. In this situation, those individuals that have a large vessel occlusion stroke, even though they're getting the best standard of care, only have a good outcome 40 to 45% of the time, 90 days later. And so in our proof of concept study, we were looking at 90-day outcomes, but we knew that it was a small study. We had about 25 individuals per arm in the study. So we chose to look at brain volume loss after the removal of the clot. And that's what no one else in the field has been able to do. We took a MRI, we took an image immediately after the clot was removed, and then we took another image at 48 hours. And that's how we measured infarct volume growth, which to us it was really meaningful as a biomarker of a potential benefit 90 days 90 days later. And so that again has given us confidence that what we've seen is real, and we're looking forward to producing it or replicating it in phase 3 study.
Well, how did that biomarker translate into clinical outcomes? What did you see on functional scales at 90 days?
Yeah, so we saw an improvement. We actually saw a great correlation between infarct volume growth during that 48 hours post the clot being removed and 90-day function. I mentioned earlier that the typical outcome for a patient with a large vessel occlusion that has the clot removed, about 40 to 45% of those individuals have a good outcome at 90 days. In our proof of concept, that number jumped to 63 for individuals that got high dose RNS 60. And we've talked to experts in the field, and movement of 6 to 8 points would be seen as really the next standard of care in stroke, and we doubled that in our phase 2 study. So there was great correlation between what we saw in infarct volume growth and 90-day functional outcome. We just need to repeat it in a larger number of patients.
Were there particular subgroups, either from time of onset or infarct size or age, where the effect of RNS 60 looked particularly strong or especially weak?
That's a good point. Good question. When we designed the phase 2 study, we wanted to match what's done with mechanical thrombectomy, which you can perform that intervention, removing the clot, out to 24 hours after an individual's had a stroke. And so that was the criteria. We essentially said, if you meet the criteria for having mechanical thrombectomy, we're going to enroll you in our proof of concept study. So we took patients in out to 24 hours since the stroke and saw this great result with 63 percent of the individuals having a good outcome at 90 days. We know time is brain in the treatment of stroke, so the earlier you can intervene, the better. So we looked at those individuals in our study, both placebo and RNS 60, that had the thrombectomy, which within 12 hours, not 24 of the stroke. And our results got even better. They grew to 72% of the individuals had a good outcome at 90 days. So this speaks to the consistency of our data. Every way that we look at it, it speaks to the benefit of RNS 60 in this clinical setting.
You've presented data showing earlier discharge and shorter hospital stays for patients on RNS 60. What did that look like in concrete terms for patients and for hospitals?
Well, I think from a concrete term standpoint, uh, how soon you can leave the hospital is a great outcome for patients. It's not something that the FDA tracks, but obviously it's something that's important to patients and their families. So those individuals that got RNS 60 left on average the hospital 6 days post um, being admitted, and placebo patients averaged 11 days. So we saw a 5-day reduction in the stay in the hospital. Again, this aligns with, if you have less infarct volume growth, less growth of that stroke post having the clot removed, when the drug is being administered, you would expect that you would be able to leave the hospital sooner. And we actually saw that patients not only left sooner, but more patients, about 40% more patients, were discharged to their house, to the home, instead of to a rehab facility. So to us, those are pretty big numbers. I think as it relates to the patient journey, at the time frame, the patient, caregivers, the hospital are all blinded to the study. So any other, I guess, learnings about that would have to remain for another time, and probably post marketing.
Stroke care is resource intensive. What's the implications of being able to shave off days of a stay, and how does that change the economics and capacity picture for stroke centers?
Yeah, you can imagine it would have a huge impact. We commissioned a firm to do an interview of insurance providers as well as hospitals on what type of impact this would have, and it was overwhelmingly positive. Is actually one of the best scorings that this group had seen with the target profile that we have for RNS60 was which was built off of even kind of a watered down version of our effect in our phase 2 study. So I think I mentioned it before, even if we saw about half the effect of our phase 2 in our phase 3, this is still really a game changer, the potential for a game changer in the treatment of ischemic stroke.
From a bedside perspective, how did RNS-60 treated patients look different during that first week compared with standard of care alone?
Yeah, I think it's hard to say because during that time frame, everyone was still blinded to treatment. But what we do know is that from a safety standpoint, there was no real difference between treatment with RNS60 and treatment with placebo. And I think another piece that I'll share is our placebo patients that received placebo in our study. Again, they had a 46% of a good outcome. 46% of them had a good outcome at 90 days, which aligns exactly with what you would expect to see in the field. I mentioned 40 to 45% is normal. So I think from a standpoint of what we witnessed, there doesn't seem to be any outliers, if you will, that would speak or that would play a role in the interpretation of the data from our phase 2 study.
And what does administration look like? Is there a treatment window? And when does the clock start? And how long does an infusion last?
So an infusion lasts for 48 hours. And in the study, we began infusion before the completion of the mechanical thrombectomy. We wanted to make sure the drug was on board right at the end of treatment. Now, given the safety profile that we've seen so far with RNS60, you can begin treatment earlier, but we felt because of the use of our infarct volume growth as a biomarker, we wanted to make sure that we captured an image post EVT and then starting the drug really right in step with that new baseline image. And I think that design of the study really benefited us in this situation. Now, what we've heard from the clinics is that because we provide the bags ready to use, there's no reformulation, there's no reconstitution that needs to be done. You're just simply hanging an IV bag. We really fit in well to standard of care. And I think it's one of the reasons why we've already been able to select 30 sites in the US for our phase 3 study because sites are excited. This is an easy study to implement. The safety profile of the drug makes it easy during the informed consent process. And then again, the benefits that we saw in our phase 2 study with reduction in infarct volume growth, you're really seeing the physiological benefit of the drug. And it's really a matter of just enrolling more patients so that we can show the functional benefit of the drug at 90 days.
And what are the primary endpoints for that phase 3 study? And how did the phase 2 study inform that design?
So the primary endpoints will be the modified Rankin scale, which is it essentially it's a 0 to 6 scale. A 0, 1, or a 2 means that you've had a good outcome. You're still independent. A 3, 4, 5, or 6 means that you're dependent. Actually, 6 represents death. That will be the primary endpoint for the study. FDA, as well as the EMA, wants to see you dichotomize your results. So, good outcome versus a bad outcome. So that'll be the design of the study. The rest of the endpoints are going to mirror our phase 2 study, and really the design is almost identical to our proof of concept study, other than the fact that we will focus on enrolling individuals that are within 12 hours of the stroke versus 24, because we saw that stronger response. That allows us to enroll less patients in this phase 3 study and hopefully collect our data sooner and be in a position to file for market authorization with FDA sooner than if we just focused on individuals that were within 24 hours of their stroke.
You're also looking at traumatic brain injury and Alzheimer's disease and other neurological conditions. What's the shared biology that makes you think this approach could extend beyond stroke?
Well, it's really the mechanism of action of supporting mitochondrial health. And again, we've got that publication coming out in an ALS model speaking to the benefits of mitochondrial structure as well as mitochondrial function. And so the healthy mitochondria play a role in really so many different diseases and conditions. That's why we feel that this really represents RNS60 represents somewhat of a platform. We're eager to move forward with our phase 3 study in stroke, but we've also got fantastic data in ALS showing that RNS60 slows the decline of breathing in individuals that are living with ALS. So there's a lot of opportunities for us, but we're focused on moving stroke forward within phase 3 and hopefully getting to that moment where we'll have the data to submit for market authorization.
And how did you come to even look at this as a potential neuroprotectant?
Well, that goes back to some of the early days of the company. The technology started off focusing on agriculture and that the delivery of a nutrient solution that's saturated in oxygen to the root zone of agricultural crops. What we learned at that time frame was that we saw an increase in crop yield, but we also saw that crops seem to avoid disease and stress, whether or not that be stress from the environment. Think about hotter temperatures or even brackish irrigation water. And Eric Russell, the founder of Revalesio at the time, said, well, if we're seeing this in plant biology, what would we potentially see in human biology? And we did some early work with Stanford. We did some work with some mid-tier pharmaceutical companies in the space and saw the potential that RNS60 had as a neuroprotected, cytoprotected agent.
Has working on RNS60 changed the way you think about what a drug is or how we should design trials in neurology more generally?
Well, I think um, when you think about what a drug is, really, it's coming down to the active ingredient in our situation. It's oxygen, and what's the indication that you're pursuing, and so we fit that criteria of a drug, and FDA regulates us as a small molecule through CEDAR. I think what it's opened our eyes to at Revalesio is the broad effect that we see with RNS60. And I know the development of a lot of drugs within the neurodegenerative space, we're looking at very targeted approaches. Now, this wasn't by design, this was really by default in what we stumbled across with RNS60. But RNS60 has this broad effect. And I think that is really benefiting us in stroke, where some of the best names in the field have really defined stroke as maybe 6 different mechanisms of injury that are happening over a 24 to 48-hour period. And so I think that's the advantage RNS60 has is its effect is so broad, it plays across that whole time span and it plays across multiple different cell types that are involved in a stroke, where maybe some of the other companies are focused more on a certain receptor or a certain target on neuronal cells, where you might only have a smaller window, maybe 4 to 6 hours, to have your biggest benefit.
How has the company been funded to date, and how far will existing cash take you?
So, great question. So we've been funded. Revalesio is a privately held company. We've been funded by family offices so far. The Russell family of the Russell financial history, so the Russell 2000 index, the Nordstrom family, and the Cumming family. And we're currently looking for investors to join us investors that are interested in really impacting uh, global health. And I think to do that, you've got to be willing to look where the unmet medical need is. And it's not looking at a new GLP-1 drug. It's not necessarily looking at a drug that was developed by AI, but it's looking into spaces like stroke or like Alzheimer's, where there's still huge unmet medical need, and then be willing to take the risk and move forward within that space. So we've found that family offices have a different risk profile, I would say, than the standard biotech investor. And that's worked really well for us to get to this stage of development.
It's been a difficult environment for biotechs to raise capital. When you talk to investors about a non-conventional therapy like this, do you sense extra skepticism, or do they approach it the way they would any other experimental drug?
That's a great question. It's interesting. I think the biggest level of skepticism that we see is about stroke, because it has been the graveyard of past neuroprotective and cytoprotective drugs. And so there is a reluctancy or a skepticism to move into stroke. And will our phase 3 data, or will our phase 2 data repeat in a phase 3 study? We think it will, because we aren't changing the design of the study, and our results were so consistent no matter what endpoint we looked at. Um, so it's I find more skepticism as it relates to the indication that we've pursued. Now, we do have an unconventional drug. There's no question about that. Um, but whether or not it's in the pharmaceutical industry or other industries, I think it's often that you really find groundbreaking solutions by looking for technologies that maybe aren't as obvious as they would be to those that are within the industry. So I think that actually gives us an advantage when you think about who's really going to bring, or maybe who has the best chance of bringing a solution to an area like stroke that has such a significant unmet medical need. And RNS60 again feels like the right compound, because again, it's got that broad effect in multiple different cell types versus that targeted effect. And we've seen it work across a very wide time window, with the proof of concept study focusing on individuals that were less than within 24 hours of stroke.
Greg Archambeau, president of Revalesio. Greg, thanks so much for your time today.
Danny, thanks so much for having me on the show. Really appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.